so excited to be in here right now and to like be able to feel my arms. I was realizing as I was coming over here today that I actually forgot in this class to do my normal second day of class thing, which I did in all my other classes. Part of the reason is because it feels like cheating, but I'll go for it anyway, just to feel like I can check off the boxes of completeness, which is where I give myself a top quiz every time on the second day of class. Like I said, it feels like cheating this time. But Emily, Nadia, Aurora, Julie, Lucas, David, Grace, Chrissy, Fisayo, Roxy, Carissa, Paige, Brooke, Jazz, Elizabeth, Miles, Tabitha, Genesis, Abigail, Ani. Yay! So a couple of general things before I get started. So the plan is that I will actually process the recording from Wednesday as well as the one from today over the weekend. I'm not planning to post a recording from Monday since A, sort of it's a little more flexible on the first day, and B, my microphone died in the middle of it, so it doesn't seem to make a lot of sense to try to recreate it. So, but the number 2 and number 3 will show up this weekend. Also, if you look at the course schedule, you will see that not next week, but the week after, there is an assignment due on pattern recognition. We don't really have a ton of information right now on it, but I do like to give you everything at least a week in advance. And Monday, which is Labor Day, where we don't have class, would be the week in advance. So I posted it. So if you look, the assignment is posted on Moodle. It's a pretty long assignment background description. There are basically 3 questions that you have to answer, but there's a lot of kind of background info listed there in the assignment. And it involves you reading 2 different primary lit papers, and so those primary lit papers are also included. So all of that is here. There are also a couple of other things I reference in the assignment, though you don't have to read them, but if you want to, either to help you out with stuff or just because you find it interesting, I linked them here. So all of this is here. Like I said, it's not totally tied into stuff we've talked about yet. I really won't tie into things until next week, but I wanted to make sure you had at least a week because it is a bunch of stuff to read. So, ta-da! So the goal for today is to finish up talking about organs of the immune system, and then to start our kind of zoom in on innate immunity. And specifically the part of innate immunity that we're starting with is the humoral part of innate immunity. Last time I introduced you to this idea that we could group different organs with relationship to the immune system. And we have organs that are referred to as primary immune organs or primary lymphoid organs, secondary lymphoid organs, and tertiary lymphoid organs. The 2 primary lymphoid organs are shown in this figure in red, the bone marrow and the thymus. And as I told you last time, they are the locations where immune cells develop. And then the plan for today was to start with secondary lymphoid organs, as this slide is titled. The secondary lymphoid organs are also shown here. Many of them are in yellow. So we have the lymph node and the spleen. Those are the big ones that most people think about, most people talk about. If I give you an example of a secondary lymphoid organ, I'm going to give you lymph node or spleen. And for my purposes, I usually think of them as pretty interchangeable, though technically there is a slight difference. There also are some other secondary lymphoid organs, like the Peyer's patches and some other specialized tissue in the intestine, like the adenoid and the tonsil, like the appendix. And the lymphatics, which are also shown on here, are also important when we think about the secondary lymphoid organs. So just for sort of purposes of notes, if you want a definition of secondary lymphoid organs, we talk about secondary lymphoid organs as the place where immune responses are initiated. That is particularly true for adaptive immune responses. So it's definitely where adaptive immune responses are initiated. So it's sort of where the immune system meets the microbes. Some people think about it as where the party happens. And so immunologists spend a lot of time thinking about secondary lymphoid organs. But to fully kind of give you the picture of secondary lymphoid organs and what's going on with the secondary lymphoid organs, I have to sort of zoom out for a second and give you a slight kind of anatomy lesson. And I will also note, because some of you who have taken anatomy will snicker at this anatomy lesson, this is an anatomy lesson as described by an immunologist. And specifically that anatomy lesson involves me telling you a little bit about the circulatory system. So if we think about the circulatory system, the first thing you should think about is that there's a really big pump that's pumping liquid. Right? Of course, pumping the liquid of the blood. In some ways, right now, you can just think of it as a thing that is pushing liquid really fast, really hard. You can also imagine it like a fire hydrant, if the fire department was coming and letting the water out. You could imagine it, you go in your backyard to like the spigot where you're turning on the hose, and you turn it on full blast. Like, heart is like this thing that is pumping liquid really hard. And then we have all the rest of the blood vessels. And again, as an immunologist, in some ways I might think about it like this. We've got a really big blood vessel attached to the heart. And then we start branching into smaller arteries, finally to arterioles, and then down to capillaries. So we go from this really big pipe coming out of our water pump into smaller and smaller and smaller pipes. And we have these really tiny pipes, and then we get bigger and bigger and bigger until we get back into the vena cava coming through the veins. So again, and you can see I've got sort of changes in things like the diameter of each of those types of, as I'm calling them, pipes, or hoses, right? All of those vessels. So I want you to just think for a second about the situation as described here. So this is really how it works. This is how you are working right now. If you took a super high pressure water source, liquid source, you know, the fire hydrant, your hose in the backyard at full blast, you attached some sort of hose to it, you attached some sort of pipes to it, and then you took that, say, garden hose, or whatever, and you squeezed it. So you decreased its diameter. As I'm basically telling you here, we have a big hose and then the diameter gets smaller. What would happen if you squeezed that water hose, say, in your backyard that's going full blast? Yeah, Roxy?
Pressure would build up.
Pressure would build up, okay? What would happen if you really squeezed it for a long time and really were having some pressure build up? So I see your, yeah, Jeff.
Might explode.
Might explode, okay? You also might stop flow because stuff isn't going to be able to go through. You can see how this is a bad situation if you are imagining your water hose. You could also imagine kind of the water spraying everywhere and destruction, right? But I just told you that's how your circulatory system works. And if you look around, you realize none of you have exploded recently. And so in fact, it has to be another piece that makes this work. There has to be another part that makes this work, and that is true, there is. And it has to do with the anatomy of the arteries. Specifically, the way that artery walls are set up is that they're permeable to liquid. And so what that means is that as the blood is going through the arteries, half of this, liquid gets pushed out. Basically like water leaves. And proteins dissolved in the water could potentially leave too. The cells can't leave, they can't go through the walls, but the liquid can. And so the idea is that you can't ever get a buildup of pressure, because when the pressure would build up, the water just gets pushed out and the water leaves, so that the cells are by themselves. I know that when I learned about all of this, at one point I was told capillaries are so small they can have one cell fit through them. And that's true, but part of it is that because there's nothing else there, like all the water and proteins got pushed out, and it's really just the cell to the point of getting to the capillaries. So this is a key thing that we think of as immunologists thinking about the circulatory system. We're excited, we've solved this one major problem. But we actually have created a second problem in having this solution. What's the second problem? Yeah, Paige.
Too much combined, and there could be empty space.
So one question could be about how much flows out in empty space. It turns out we actually don't end up having issues with that in a healthy individual. And one other really nice thing, to be honest with you, is because the liquid and whatever's dissolved in the liquid can go through the walls, you're also basically bathing your tissues in nutrients. As the blood comes by, it's kind of giving everybody a bath of nutrients as that liquidy nutrient water goes through. But the problem is that basically you have all this water in your heart. Technically, if any of you have taken anatomy types of things, the name for the water that's getting pushed out, the technical term is the interstitial fluid. So if you've heard of the interstitial fluid, that's what I'm talking about. So this is great. We pushed all this water out. Our blood vessels have not exploded. But now we push this water into our tissues. And if we just kept it there and let it sit there, we would turn into Jabba the Hutt. We would just be a big blob. We can't just let the water sit there in all the tissues. We somehow have to get it back so that it can get back at least by the time it gets to the heart. It might come back at other places. You don't have to get super stressed about it, but it has to come back into the blood at least by the time it gets to the heart, so that it can get reoxygenated and all that good stuff, and so that we can get more nutrients, and so that you are not a blob. And so the way that this works is that we have a second system. And the second system is a collection system for the liquid. So this system is sort of totally linked with the circulatory system. It's really hard to imagine this as like its own separate thing, because what it's doing, at least in one level, is collecting the liquid to help make the circulatory system work. So it's collecting the liquid back up so you're not a blob, and giving that liquid back to the circulatory system. That system that is picking up the liquid is the lymphatic system. So basically what we're talking about here is the lymphatics. And you can see sort of an example of the lymphatics here. So we've got arteries. You can see that liquid might be leaving those arteries in these white arrows. We've got our veins, and we've also got sort of these lymphatics that are picking up the liquid, bringing it back into the circulatory system. You can also see these lymphatics in this slide. So here you can see this sort of whole system of little tiny capillaries coming into bigger and bigger vessels, all collecting the liquid, bringing it back to the circulatory system. There are a couple of unique things about the lymphatic system. One of them is that there's not a pump. There's no heart. There's no sort of specific muscle that's pushing all the liquid through the lymphatic system. The lymphatic system is set up a little bit differently. Instead, the lymphatic system is basically open tubes around your body where the goal is that liquid could get pushed into them. So if, as you have that liquid just around in the tissues, if it goes by the open tube of the lymphatics, it flows in. And so you can see at the top, we just sort of have these open capillaries drawn. You can also see in this view from your textbook, we sort of have these open vessels drawn. And it's basically just collecting whatever comes by. Then things sort of move from the small vessels to big vessels. It's officially lymphatic drainage. And all of those vessels have valves on them, have little gates, so the liquid can only flow one way. So it can only flow forward in the direction you want because there are vessels preventing, or valves preventing flow in the other direction. But like I said, there's no heart, there's no thing to pump all this around to create pressures. The way you actually create pressure, the way you push liquid into the capillary in the first place, and then move it around your body, is by body movement. And so as your body moves, that's pushing the liquid around and potentially pushing it over one of these lymphatic capillaries. And so that collects up the liquid, collects it back into your circulatory system. The liquid can only move one direction because of valves on the lymphatics. And so it's really this nice big, water collection system, keeping you from being a blob. But it also has another function. And in some ways, it's sort of like, a clever thing, clever taking advantage of thing, in that we're basically getting liquid from the entire body. We're collecting it up. And so if we set things up correctly, we can use that as a way to do surveillance and make sure there's nothing bad in the entire body. So it gives us a way to check what's in the body too. And so we've got these lymphatic vessels that are picking up whatever liquid is in the body, but they also, as you can see here, can pick up microbes. They also actually can be highways for cells to travel on. So cells can travel there, microbes can travel, and you also have all that fluid and other type of stuff. You have random proteins. And so whatever is sort of in the body, in your tissues, is just flowing into your lymphatics. You're moving it around with body movement. People who are bedridden sometimes have to be flipped and things, and it's because you're actually trying to not let lymph pool in different parts of their body. And but the idea is that we have these little organs every so often in this circulatory system that let us check what's in the liquid. Are there any microbes in that? Are there any problem things in that liquid? And so we made this we have the circulatory system, and then at different places along it, we have checking sites where we can check things out. Those are the secondary lymphoid organs. Most specifically, those are the lymph nodes. And so you can see the lymph node is sort of collecting the liquid, potentially any microbes, cells, collecting a whole bunch of stuff, kind of pushing it together nicely so that that's a good place where we can do immune surveillance. And you can also see this system here. So here you can see the heart with all the cells coming out. Those cells can go to the lymph node if they want to do surveillance, if they want to kind of see what's going on. You can see those cells can then go back to the heart through, they can either go through veins or through lymphatics, but you can see basically the lymph system dumps everything back into the heart. So we join up all the parts of the blood that separated by the time we get back to the heart by dumping everything back in. You can see this here as well. And so you can see, we've got arteries and veins, but we also have the lymphatics in the yellow line that are dumping everything back into the blood. So it all comes back together again. Yes, there are places where it could also join up earlier, but we're not going to deal with that level of anatomy right now. And so one of the reasons why this is really useful is that you can imagine being, say, an adaptive immune cell. Now imagine because I told you secondary lymphoid organs are especially important for adaptive immune responses. You are an adaptive immune cell, a lymphocyte, that is specific for tetanus bacteria, Clostridium tetani. And maybe even imagine I just stepped on a rusty nail. I got some tetanus. Okay? You're that T cell. You're going to start right here in my body, and you're going to be like, do you cell have tetanus? Do you cell have tetanus? And you're going to go all the way across my head, and all the way, right? Think about how long that would take before it got to my foot where the rusty nail was. And think about how much time the bacteria would have with sort of no checking. And so it doesn't make sense for the adaptive immune response to be actually going cell by cell through the whole body. Instead, what the adaptive immune cells are doing is they're actually looking at regions. They're kind of like, does this half of the room have it? No, okay. Does this half of the room have it? Oh, somewhere, so I guess I better look more closely. And the way that that's working is that we have these secondary lymphoid organs that are placed all around the body, the lymph nodes being sort of the key ones. And each secondary lymphoid organ is responsible for one region of the body. It's the place where the liquid from that region goes. And that means the adaptive immune cells only have to go to all the lymph nodes or all the secondary lymphoid organs and say, like, did your region have it? No, okay. Did your region have it? No, okay. Did your region have it? No, okay. And so those cells can actually look in the whole body much faster. When they get to a particular region and they go to a particular lymph node, then they can say, okay, now I actually have to go look more closely through that region. But we've sort of identified which area is sort of the area where the microbe is. It's really sort of reduced the work. And so this lymphatic system is doing a lot of things. It's collecting our liquid, it's reducing the work of the cells, it's making us have a nice surveillance system. Kind of a very nice setup here. So you've got lymph nodes throughout your whole body, which, as I said, are sort of the most famous secondary lymphoid organs. So like you have lymph nodes here, for example. This lymph node is responsible for this portion of my head. It's draining all this portion of my head. This lymph node is draining this portion of my head. There's a lymph node behind your knee. It drains the bottom half of your leg. There's a lymph node in your groin. That's the top half of your leg. There's a lymph node in your armpit that does certain parts of your arm. So there are lymph nodes basically all over your body, and they each have a place that they're draining. They're usually referred to as the draining lymph node. When your lymph node is like active, it's the one that's draining the infection. We call it the draining. So we have these lymph nodes throughout the body. You can see lots and lots of lymph nodes all over the body, shown here, which are kind of these little larger circular areas that drain different parts of the body, which is basically just a way of doing surveillance on that section, allowing the adaptive immune cells to find their microbes there instead of having to go look everywhere randomly. I mentioned last time that I was a little sad about you guys missing the lab where we look at blood cells under a microscope, and that I was happy to do that if somebody wanted to set up a time. The one other thing that I'm sad that you missed is that I also would do a mouse dissection, so you could actually see lymph nodes and see us bleed and see all of the immune organs in a mouse. And I think at some point this semester, Dr. McKittrick is going to have some mice that she's not going to need. And so I can also do some organs if you want to see lymph nodes and you want to see most of the organs I tell you about today, and even the primaries, the thymus and bone marrow, I can get out of the mouse and show to you. So when I know more about when I have mice, or if that's something that anybody's interested in, let me know, and we can make that happen. So we've got all of these lymph nodes as our big secondary lymphoid organs that I've been telling you about. If you look closely at a lymph node, it does have individual parts within it, so there are different sections in the lymph node. We're going to care about those more later. We don't really need to care about them right now, but yes, there are different individual sections. You can actually see some of them under a microscope really easily. All of our lymph nodes have arteries and veins coming in and out, as well as lymph vessels coming in, which are the afferent lymph vessels, and lymph vessels going out, which are the efferent lymph vessels. So cells have a choice. They can come through the arteries and veins, or they can come through lymph vessels. Liquid microbes, they just come through the lymph vessels. And we've got all of these different locations in here. When I was actually taught first to get lymph nodes out of a mouse, there's usually some connective tissue around here as well. And I was actually taught that you look for the place where a bunch of vessels come together that looks like snot. Because of all the connective tissue. And honestly, like, if I show it to you, it's a pretty nice definition. But there will be this nice, like, white little sphere, like, perfectly white sphere, and that's really the lymph node that's at that specific location. The other kind of really important secondary lymphoid organ is the spleen. The spleen is sort of in the back of your abdominal cavity. You can sort of see it is shown right here on this person's their left, your right, when you're looking at them. And the spleen also has a really important like structure within it. Some of the parts of the spleen are exactly the same as the parts of a lymph node. That's part of the reason why I think of a spleen as a really big lymph node. The difference, or the sort of unique thing about the spleen is that the spleen, you can kind of imagine, is the lymph node that checks the blood in general. So it's like the lymph node checking the blood, the actual blood going around your body. And it has a second function, which was red blood cell recycling. So it has some extra parts for red blood cell recycling that we're not going to care about. A big reason why I mention that is because if you actually look at this in an organism, the spleen is red and a lymph node is white. Because the spleen has red blood cells in it and the lymph node doesn't. But again, I think of the spleen as a giant lymph node, and it really is the lymph node that's checking on the blood. In some parts of the body, we also have other types of secondary lymphoid organs. So in the gastrointestinal tract, there are a number of tissues that are known collectively as the GALT. And GALT stands for gastrointestinal, that's the G, associated lymphoid tissue. And so it's gastrointestinal associated lymphoid tissue. Some of it is just kind of some specialized things in the intestine that are actually kind of hard to totally distinguish, and I'm not going to get like super into it right now. But actually in the small intestine, there are specific little areas where you have like a lymph node in the wall of the small intestine. It's called a Peyer's patch. And so you can see like suddenly in the middle of the GI tract, there'll be like a little lymph node area, like in the wall of the GI tract. You can see it here and here. And you can even see this is a mouse's small intestine with these little white bumps on it. And those are those Peyer's patches, which are an example of gastrointestinal associated lymphoid tissue. And we have a lot of sort of GALT types of tissues, and that's kind of important because you're eating a lot of potential microbes, and you really need to be looking for microbes in things you're ingesting. There are some other types of secondary lymphoid tissue that you could give them lots of different names, but some ways that people talk about them sometimes are MALT, which is mucosal associated lymphoid tissue. Sometimes they talk about BALT, which is bronchoalveolar. Sometimes they talk about NALT, which is nasal. All of these things are sort of closely related. They all can be specialized structures in some of these individual locations. Things like the tonsils and the adenoids would be examples, but there are also some others, just like in the wall of the lung and things like that, that we're not going to get super into. Understanding what's going on in some of these types of tissues is, one of those areas that a lot of immunologists are finding really hot right now. So I've told you about primary lymphoid organs. I've told you about secondary lymphoid organs. And so I also have to tell you about tertiary lymphoid organs. And tertiary lymphoid organs are pretty easy to define. So I'm going to give you a definition of a tertiary lymphoid organ, and then I want you to give me an example. A tertiary lymphoid organ is a place where there's an immune response happening. So it's like a place where there's a microbe or something that has to get dealt with, that the adaptive immune cells, once they're turned on, go deal with. So what's a tertiary lymphoid organ? What is it, Paige? Anything. So anything can be a tertiary lymphoid organ. A lot of times we'll say, like, if you have a cut and you have like a wound, we'll say that your skin at that point is being the tertiary lymphoid organ, but we might say it's not like always actively doing it. But wherever you kind of have something, an immune response that has to happen, it's a tertiary lymphoid organ. And so anything could be a tertiary lymphoid organ. Really pretty straightforward. Yep.
Wait, so in terms of semantics, does it become a tertiary lymphoid organ when it has to have an immune response, or is it like everything is consistently a tertiary lymphoid organ?
Yes.
Okay.
It is always, it is a tertiary lymphoid organ when it has to make a response. But there are some things that don't really ever not make responses.
Okay.
Is the answer to that question. That's why I was like snickering as you were starting to talk. And so that's why if you actually look back at this paper that I showed you, you can see some of the primary and secondary lymphoid organs having a lot of immune cells sort of per gram, but you can also show that they show like everything on here, and it might potentially having immune cells, because that might be a tertiary lymphoid organ in that situation. So you can find immune cells in a whole lot of places. I can tell you from painful experience that you could even isolate them from a lot of different locations if you want to do studies on them. But some of them I would never, ever, ever recommend trying to get them from, because it's really annoying. So now I'm actually going to switch gears into thinking about innate immune responses. Oh, sorry, Emily.
Sorry, this is just a
Go for it.
question. So blood and lymphatic fluid both are going through lymph nodes, and they kind of like process both of that.
Yeah. So as this is going to be a much later thing,
Okay.
but blood is going to go through lymph nodes, but sometimes the blood will just kind of pass through,
Okay.
and sometimes an immune cell in the blood will be like, whoa, I think I need to stop here.
Okay.
And it will stop, and it will actually move into the other parts of the lymph node, into some of the other portions.
Okay.
So it's usually going through when it circles around, but it may or may not want to stop.
Okay, but like the lymphatic is more like closely looked at, almost like.
Yeah.
lymphatic fluid.
So the blood vessels are actually specialized, so that the cells can kind of figure out whether they need to be there or not.
Okay.
Is this the short answer for right now?
Okay. And then like the B cells, is there like do they switch around too? Because like what if you had a bacteria in your leg, but like your one B cell for it was in like your head?
Yeah. No. So this is true of all the adaptive immune cells. So B
Okay.
cells and T cells.
Okay.
Totally agree with us. Yeah. So we're going to talk about specifics of innate immune responses now, and I'm dividing up innate immune responses into humoral and cellular. If you remember, humoral is kind of where proteins or molecules are really important. Cellular is where cells are really important. When I think about innate immune responses, I've mentioned before that I think about kind of the first 96 hours after an infection. Even that can be kind of divided up. there are some things that are active immediately, like time 0, time 1 minute, you know, as soon as that microbe comes into your body. And that's really important because those microbes divide so quickly that if you had a gap of time at all, that would be a problem. Other things take a little while to get turned on. So when we look at an innate immune response, you can see that your textbook kind of divides it into like 0 to 4 hours and 4 hours to 4 days. But like there's like the really early part, that first 4 hours, and then after that. And the proteins of the innate immune response are the things that are working immediately, like time 0, time 30 seconds, time right away. And so that's why we separate out the humoral versus cellular, because it's the proteins that act immediately. The cells might take a couple hours before they're really ready to go. The great thing about the proteins is that they are pre-formed. So those proteins, you already made them, you already transcribed and translated, you already have them circulating in your body. So they're already there, and immediately if there's a microbe, they act. You don't have to get ready at all. And so with the cells, you gotta do a little getting ready. And so what can happen is that you can have a pathogen come into the tissue. These pre-formed proteins can act against it. I can reach, I'm tall. Pre-formed proteins can act and reach against it. Sometimes that's good enough, and those proteins actually get rid of the microbe and you live happily ever after, and you probably never even noticed there was anything going on. And so these proteins are pretty great. They do a lot for us. Sometimes they don't do everything, and then we have to wait around for those cells to act. But sometimes those proteins have done the whole job for us. Even if they haven't done the whole job for us, they've probably done the job of keeping the microbes from reproducing exponentially, keeping the number of microbes down, so that when the other immune responses finally start up, there aren't as many individual cells to deal with. Paige, your question.
So when it says the induced innate, that's just because the cells have to be made, like they're not already just sitting there.
So the cells actually have to make some changes in terms of things like transcription, translation. That's going to be kind of a next week problem.
Okay.
So really what I'm showing you here is why we're separating the proteins and the cells, and we're going to spend the rest of today with the proteins. The other thing that's sort of unique about the proteins to think about is that these proteins are particularly good for microbes that are extracellular, that are outside of your cells. These proteins don't necessarily do much if the microbe is already inside your cell. It's kind of like protected and shielded by being inside your cell, by your cell's membrane. But if that microbe is outside of the cell, these proteins are going to be able to work on it really well. There are lots and lots of types of these innate immune proteins. Some of them are just sort of generally talked about as antimicrobial peptides. And this is where I was having that debate with my former student that I mentioned to you. Because there are antimicrobial peptides, they immediately attack microbes, but they are made in large amounts in barrier organs. So one of the things that makes barrier organs special is they make a lot of antimicrobial peptides. And so are the peptides barrier or the peptides innate? Like, one could debate that point. But so what I want you to see is that many of those barrier organs I'm telling you about, they do have different types of chemical defenses, mechanical defenses, things like that, but they generally also all have some of these antimicrobial peptides. So they're making these innate immune proteins sort of at that particular time. One other thing that I will mention is when we think about innate versus adaptive immune systems, I should have said this last time. I told you on the first day we're thinking about the immune system of mammals. And that's really because mammals, or most vertebrates, have adaptive immune systems that are really cool. The adaptive immune system is actually a unique feature of certain types of vertebrates. Innate immune systems are basically in every organism. So every organism has some kind of innate response. There are antimicrobial peptides, like I'm showing you here, that are in barrier organs, that are also in plants. There are antimicrobial peptides like this that are also in fish. They're also in frogs. Basically, if you can think of an organism, it actually has some of these antimicrobial peptides. I know at the place I taught before Drew, there was a person in the chemistry department who worked on antimicrobial peptides of fish. And she totally was like, look at my chemistry. And I'm like, it's not chemistry, that's immunology. What do you mean? And she's like, no, this is chemistry, but these are these proteins that kill bacteria. And I was like, yeah, they're innate immune proteins from the fish. So anyway, it could be kind of an interesting situation. But yeah, these antimicrobial peptides exist a lot, throughout life. Again, there are tons of antimicrobial peptides. I can't tell you about every single antimicrobial peptide that exists. And so I want to tell you a little bit about a couple of kind of standouts or really important ones, but know that there are tons of them. They all have unique special ways that they work. They're generally made by barrier organs. And you'll see this in one of my other examples as well. But one thing that we should be aware of is that sometimes just the presence of a microbe doesn't hurt you. Sometimes there has to be some process the microbe is doing that hurts you. So some of these antimicrobial peptides actually stop the microbe from doing the thing that hurts you and leave the rest of it alone. I think people often think about this as just it's all about killing the microbe, and some of it can be much more interesting and trickier about how these antimicrobial peptides deal with the microbes. So one of the examples I want to just mention quickly here, I don't have a slide for it, is one called lactoferrin. Lactoferrin is a really important antimicrobial peptide in your body. You see it has fer in it. What does that remind you of or make you think of? Here's some whispers of iron. And there are correct whispers of iron. Microbes, like all other cells, all other organisms, need iron to survive. And so lactoferrin is basically a protein that you have in your body that binds all your iron and hides it so the microbes can't have it. And it's an iron sequestration protein. And it just hides the iron so the microbes can't have it. In fact, there's a whole evolutionary fight about this. It's called, cool name, iron piracy. When the microbes try to steal your iron, we call it iron piracy. So that's not what people often think about when they think about antimicrobial peptides. The mechanisms are really diverse. One, perhaps the most famous antimicrobial peptide that exists is one called lysozyme. Note the name of this peptide carefully. Note that the end of the word is zyme, like enzyme, and it is in fact an enzyme. The reason why I mention that is that later in the semester, we will talk about an organelle, the lysosome, S-O-M-E, instead of Z-Y-M-E. It's an organelle. So note lysozyme is the enzyme, lysosome is the organelle. And when we think about different types of prokaryotic organisms, many of them have a particular molecule in their cell wall called peptidoglycan. This is found in gram-negative and gram-positive bacteria's cell walls. It is not found in your body anywhere, or at least in any of your body parts. It's also not found in fungi, it's not found in viruses. It's only gram-positive and gram-negative bacteria, and it is a polymer with a very particular structure. One of the chemical bonds in peptidoglycan structure is called a beta-1,4 linkage. It's the way that one part of the structure is bound together. And the way that lysozyme works is that lysozyme is an enzyme that specifically breaks the beta-1,4 linkage in peptidoglycan. And so here you can see a gram-positive cell wall, which contains some peptidoglycan, these purple circles and blue hexagons. Here you can see a gram-negative. We've also got some peptidoglycan. And here you can see this very biochemically accurate view of lysozyme, and it's breaking the linkage between those different subunits, which is the beta-1,4 linkage. So now the lipid bilayer is exposed, and basically what can happen is this cell can be lysed really easily with osmotic shock. And so this cell is likely to lyse because it doesn't have the cell wall to protect it. The other thing that's really important is that you can make all the lysozyme you want in your body, because it's not going to hurt any of your cells. Your cells do not have peptidoglycan. And so lysozyme is not going to harm your cells at all. And so this is a key feature of pretty much all of our antimicrobial peptides. And it is a general principle we think about in immunology is how is this involved in sort of distinguishing foreign things from our cells? And here it's really easy. We have an enzyme that's attacking something that's only in other cell types, not in our cells. So it can't hurt us at all, at least our personal cells. And so there are lots of antimicrobial peptides that have some kind of interesting specificity in this way. The other big group of antimicrobial peptides, if you were kind of to put them into groups, is a group that are known as the defensins. There are lots of types of defensins, including the alpha defensins and the beta defensins. I'm just going to call them the defensins for our purposes. And these are, like I said, another group of antimicrobial peptides. A couple of things to notice with this slide telling us about defensins is first about where these defensins are made. And so what you can notice is that if we look at all of these different defensins and we look at the tissues where they're made, what do you notice about many of the tissues where these defensins are made? Yeah. What were you saying?
Oh, they make fluid?
Okay, they make fluid. Yeah, Jazz.
They're part of the digestive system.
Lots of part of the digestive system. What else? Yeah, Abigail.
They're barrier organs.
They're all barrier organs. They're all in contact with the outside world and likely going to be, places where you might see a lot of microbes. So you're going to be making a lot of defensins there. One place where a lot of people think about defensins is that if you look at your GI tract, you have sort of in your intestine these big long villi and these deep little crypts where stuff can get down in, and that crypt makes tons of defensins. That crypt is like super full of defensins. The other thing to notice, one thing that you will notice is that some of them are made by like neutrophils or other innate immune cells. Again, this is sort of that place where the line is a little fuzzy between the different aspects. The other thing to look at is what it says here about regulation of the synthesis of these defensins. And so what do you notice about regulation of these defensins in general? What do you see a lot of in that column? Constitutive, yes. You see the word constitutive a lot. Do you know what the word constitutive means?
Is it in there?
Yeah, Miles.
It doesn't need to have a trigger in order to be produced.
Mean is on all the time. You don't have to turn it on. It's basically permanently turned on. It's turned on by default. I often think about it as the default of if you don't tell it to do something, this is what happens. It's constitutive in biology. So all of these are just things that are being made all the time in those organs, which makes sense because they never know when they're going to get a microbe to show up. So they should probably have some around all the time by default. I'm going to tell you about defensins and how they work on the next slide. Though I realized while I was talking about this, I forgot to tell you one other fact about lysozyme, which is lysozyme is in a lot of these same barrier organs. It's in a lot of places. You can find it all over the body. But there are two places where actually there's a huge amount of lysozyme. And so when researchers want to study lysozyme, these are the things that they try to collect, which is spit and tears. And so your tears are actually full of lysozyme. And it makes sense, you're trying to protect your eyes from infection in the eye, so you have a lot of lysozyme in the eye. But yeah, and so in fact, when people want to study lysozyme, they usually try to collect tears. So that was my random lysozyme fact. But now back to defensins, and how defensins work. I told you that lysozymes are an enzyme that are breaking down beta-1,4 linkages, and in peptidoglycan, it was really easy to think about how that would affect microbes, but not our cells. Defensins are charged peptides. So these peptides have a positive charge, a really strongly positively charged peptide. And the way that these defensins work is that this charged peptide can actually insert into a plasma membrane. And so you can see that insertion happening here, and that disrupts the plasma membrane and induces a pore in the plasma membrane. And so this is how a defensin works. And I very much remember the first time I learned about defensins being kind of frustrated by them, because I thought, well, that seems like a good way to kill a cell. I'm on board with that. But I don't see how it doesn't kill our cells. How is it that these constitutive on all the time defensins are letting me be alive? And it turns out there are kind of there's sort of two things that are a part of it. The most well understood and the most famous actually has to do with phospholipids. So remember that phospholipids have head groups, and those head groups can have charges. Our cells are really organized with those phospholipids. They make it so they're known, so they make sure that there are no big negative charges on the outside of the cell. That's important for like a lot of ion transport, nerve transduction. It's important for a lot of things that we don't have time to deal with right now. But we basically organize. And so there's no negatives on the outside for the positive to interact with. Microbes don't organize their phospholipids. They just have pluses and minuses any old place. And so there are some negatively charged phospholipid heads for the positive defensins to interact with. And so I remember being very annoyed by defensins for a very long time because I couldn't imagine how they could be specific. The other thing that sort of is out there, and I don't totally understand it, is the defensins seem to care like exactly how round the membrane is. So in like our cell, it's like this, and if you have another cell that's like this, it kind of knows like that's like a tighter curve than this. Like it somehow can actually sense like the arcness or the angle of it. I don't totally understand it, but there are papers out there that kind of say that there's something about that going on structure-wise too. So if you're very worried about your cells, don't be worried about them. We don't totally understand it, but you're not going to die of defensins. The one other example on this that I just want to mention is one called alpha-2 macroglobulin. And again, the reason why I'm mentioning this is to show you kind of the diversity of ways that these peptides can be antimicrobial. Many microbes cause disease because they make a protease that disrupts something in your body. They make a protease that hurts you. The rest of the microbe does not hurt you at all. But this one protease that the microbe makes is a problem. And so alpha-2 macroglobulin is really interesting because basically it just tries to get rid of proteases and doesn't deal with anything else. So alpha-2 macroglobulin actually has this little peptide section that's shown here that says bait, that is a little bit of protein that it looks like what a protease might want to attack. And obviously this has specific for certain proteases. And that's important because you know your body also needs proteases. But you have this bait for some microbial proteases. We've got microbial protease. If the microbial protease attacks the bait and tries to degrade alpha-2 microglobulin changes its structure, conformational change, and traps the protease, so the protease can't hurt you anymore. And so you can see it's just this sort of trapping mechanism. Doesn't hurt the microbe, the microbe keeps on growing its normal way, but the protease, the thing that's causing damage to you, is not active anymore. And so, again, it doesn't have to just be killing the microbe. There are lots of different mechanisms that it seems like some of these proteins are working by, which I think is pretty cool. So, if another immunologist looked at my course schedule and saw the words humoral innate immunity, they would be like, oh, I know what she's talking about. Because there's one other type of antimicrobial peptide that immunologists think about. And it's like a big deal. And so for the rest of the time today, and for some time on Wednesday, we got to talk about this other set of proteins. So it's kind of a big deal. These proteins are known collectively as the complement proteins. Jazz heard me complaining yesterday. No, there's actually two ways to spell complement. Like whether you're adding to someone or whether you're giving them, like telling them nice things about themselves, technically spelled differently. This is the spelling is the adding to things spelling. So I was complaining yesterday, and I was saying to Jazz, when I was in grad school, when I was an undergrad, oh my gosh, did we make fun of complement. And we would complain about complement so much. Complement is the most boring part of all of immunology. Why do we study it? Why do we have to? When we were TAs, it would be like a thing of like who draws the short straw, and actually has to be the one who like teaches about complement, because we all hate complement. And a lot of immunologists do kind of have this negative idea of complement. Part of it is because it was described a really long time ago. It's one of those things that immunologists did a really long time ago, and so the naming is wackadoodle. And part of it, there's other things that I'm like, yeah, that's so much cooler, so many more bells and whistles. But the thing about complement, and the reason why it's really key that we spend some time with complement, is because complement is this really key part of the innate immune system that you actually can't survive without. But also, one thing that's really interesting is if you actually, and again, when I was thinking about complement as a grad student, I was like, oh my gosh, I'm gonna fall asleep when I talk about it because it was all described in like 1920 and no one even cares. So this is actually a paper that I get emails from certain journals. This is a paper I got in an email yesterday. This is from 2012, but it's still a little bit older. And what people are finding is that there are more and more diseases that nobody thought were immunology diseases that all happen because of complement. And so this shows you, this paper that I saw yesterday said that complement was the major factor in osteoporosis. This is actually showing these little whites. This is an eyeball. These little spots are actually complement in the eyeball that are leading to macular degeneration. Complement has been shown to be super important in both osteoarthritis and rheumatoid arthritis. Complement's really important actually in neuroscience for synapse pruning. We just keep finding more things where complement ends up being the really key feature. And so I know that my past self totally underestimated complement. And so we should not underestimate complement because it is really, really, really important. In general, we're going to learn a whole bunch of different events and and a whole bunch of different parts of this process of complement. You can see complement listed here on the top. You can also see sometimes when immunologists, if they were trying to be like using shorthand, will write C prime, and that stands for complement. So if I ever by accident write C prime in something, I mean complement. And we usually talk about complement fixation when we talk about complement. And this is one of those things about it being old-timey, and everyone just, and it's getting described a long time ago. They made up this name, and it's dumb, but it's what we use because it's the name. Complement fixation basically means complement gets stuck to stuff. Or more specifically, complement is attached to something. And so if you attach complement to things, it's said that you are fixing complement. Or if you are going to start attaching complement to things, you say you're doing complement fixation. In the general idea of complement fixation, we're going to see some complement proteins, like this blue oval. They have terribly descriptive names, like C3. And we'll start to see the names of them and go through the process of it in a little bit. But we've got a bunch of different proteins like C3. In every case, or in at least most of the cases we're going to see, these cases we're going to care about, what happens with complement is that our complement protein, like C3 shown here, gets cut by a protease. There's some special protease that cuts the complement protein, and when it's cut, it goes into two pieces. And one of them's A and one of them's B. And we're actually not always going to even care about A versus B because some of the naming is so weird. But there's always pieces. One piece floats away, and one piece gets attached. And so you can see the idea is that we proteolyze this protein, this complement fragment. We cut C3 with our protease, and one of the pieces gets stuck to the microbe. So we fix this complement onto this microbe. And there's another piece that might go off and do something else. That's the thing you're going to be looking for in complement. So here's the reason why, or one of the parts of the reasons why everybody hates complement. With complement, there are 3 different ways complement can get started. So I think of these as 3 different initiation pathways. So there's sort of 3 different ways you can start complement. All 3 of them go to like a common middle place. So we're going to have, I'm going to be like telling you this protein, then this protein, then this protein. We're going to end up at the same place, no matter what we started with. And so you can see we've got 3 different ways of starting. We come together into this common middle area. There are a few different things you can call it. I'm not going to get super stressed about what we call it. I usually just say the proteins who are there. Then there are 3-ish ways you can kill the microbe. Or not necessarily kill the microbe, but do something about the microbe. And so one of the things that when we start talking about complement cascade, you're going to be thinking about, am I in initiation or am I killing microbe? Which part of this whole business are we talking about with any given step? And we might think, well, am I at this initiation, or this initiation? Or am I down here at which of these? So you're kind of going to have to identify where are we. And I'm obviously going to give you hints about what tells you about where you are at different points. The bottom parts are often referred to as the effectors, or the effector function, or something like that. Immunologists love the word effector and effector function. It was actually kind of funny because I didn't realize that, like, I had been in immunology long enough that, like, that was just a normal word to me. And at one point I said it to a non-immunologist, and they're like, what is this thing you keep saying? And I like forgot that it wasn't a word everybody knew. So effector is this word you're gonna hear a lot. And effector means like how you do the thing, how you kill the pathogen, how you have your effect. It's like the actual like final job part. So that's how we're gonna get rid of the microbe. So we're gonna have 3 initiation pathways. They're gonna come together into a central sort of part of the pathway, and then we're gonna see branching into different effector functions. You could have any initiation pathway go with any effector function. In fact, usually you'll initiate and you'll do all of the effectors or something like that. But you could kind of mix and match it anyway. So this is sort of your textbook's big picture overview of this. And this is the big picture overview of this from a different textbook. But again, same basic idea of how this works. So the way that I like to talk about complement is I pick one of the initiation pathways, and I talk about that whole initiation pathway and get us to this central part. And then I talk about the central part and how it works. And then I talk about the effector functions. And then I come back and say, or you could have started these other places. And this is where they join up. So that's kind of how we're going to do this. I'm going to talk today a little bit about one initiation pathway. So no, yes, there are other ones that I haven't gotten to. I'm going to start with one of them. The one of them I'm going to start with was the first one that we discovered, the first one that I learned, so the one that was originally thought about with complement. We now know this pathway that I'm going to tell you about first actually evolved last and is like the newest one we had. But it just so happened that it was the first one we discovered, so that's all we go with. And it's referred to as the classical pathway. So we're going to start with the classical pathway. In the classical pathway, we start out with an antibody binding to a microbe. You can see the antibody here as this Y-shaped protein. You can also see sometimes you get antibodies together in a group. We don't care about that right now. We'll care about that later. The moral of the story is the antibody bound to the microbe because the microbe had something foreign about it. And that is what's going to start the classical pathway. There's something really important about this step with classical pathway. Antibodies, which as you can see are the key part of the beginning of the classical pathway. Antibodies are part of the adaptive immune system, not the innate. And so the classical pathway actually requires you to have adaptive immune responses going on. The classical pathway, that's why it was the latest one to evolve and the one that sort of is sort of the fanciest one, is it actually requires an antibody, requires an adaptive immune response. So you can't do it necessarily all the time. In fact, the way that it was discovered was people found there were these interesting proteins that made antibodies better. They complemented them. So that's why they called it the complement pathway. We'll talk much more about the structure of an antibody later, but now you can see it's a Y-shaped molecule. So there's 2 arms and a tail. Once the antibody binds in the classical pathway, so my classical pathway starts with antibody binding, and immunologists love to say AB for antibody. Something is going to bind to that antibody, specifically to that tail part that's now hanging out up in the air, waiting to have something bound to it. And in fact, it's probably sitting at a special geometry because of how it bound. So you've got sort of these tails that are now in a special geometry. And that can be bound by a specific protein, a specific binding protein. And that specific binding protein is called C1. So the antibody is recognized by a protein called C1. The first complement protein, C1. This shows C1Q, C1R, C1S, I'm not going to distinguish the functions of the different parts of it. I'm just calling it all C1. You can see your textbook's version of C1 as a cartoon on the left, and you can see C1 and under an electron microscope on the right. And you can see that is legit what it looks like. People often describe C1 as kind of looking like an upside down bouquet of flowers. And that structure, that upside down bouquet of flowers, this of it is actually really important to its function. So you want to sort of recognize that structure. And the idea with C1 is that our upside down bouquet of flowers can bind to antibody tails. And so if the antibody tails are sort of in the right geometry, now C1 will bind to them on the pathogen surface, and that starts this whole process. When C1 binds, it makes a conformational change, and it now becomes an active protease. And this is again with that QRS business that I'm not getting super into. We now have an active protease. And so now there's a protease that was around that wasn't around before. It wasn't active before. C1 was always around, waiting, ready to go. But now it had found something to bind to. It's an active protease. And as an active protease, it can cut another complement member. Because remember, the key thing about complement members is they get cut by a protease. So we just turned on the first protease. So now we can cut something. So where you learn that immunologists don't know how to count. In a perfect world, the thing it would cut would be called C2. But it's not. It's called C4. So now we've got C1 attached. I'm going to show you this in the next slide in a second, but you'll see why I'm kind of... Some of C4 is going to get attached, and this part of C4 is going to become an active protease. And it is going to be able to cleave the next thing in the process. And the next thing in the process is called C2. Different people do some weird naming with C4 and C2. So basically, I'm just going to kind of say like C1 goes to C4 and then C2 and not, again, zoom into some of the really picky details on it. And so you can see your textbook kind of puts them both together pretty quickly in that we've got C1 binding to the antibody tails, and now C4 and C2 both end up cleaved, they both end up attached. So you have sort of part of C4, part of C2 now attached to this microbe. So you cut them, part gets attached, part goes away. You cut them, part gets attached, part goes away. Once you have this leftover piece of C2 and this leftover piece of C4, this combo together is an active protease. And it cleaves the next thing. And the next thing that is cleaved is C3. And so you can see that your book set tells you this is called a C3 convertase. That means it's a protease that cleaves C3. You see there's another one of those old-timey words that the complement people love. Convertase means it's like the protease that cleaves C3. So we get our C3 convertase. And C3 is part of this middle section. So now we're at the part that's common to all aspects of complement. So our other initiation pathways are basically other ways that we can get C3. We can get a protease that's going to make C3. And the effector functions are sort of things that are going to happen after C3. Technically there's one other partner, which is C5. It's going to come in here, but we get C3 and C5, and then we branch out from there. And so, again, you can see here is C1 binding to antibody, turning on C2 and C4. Now C3 gets cleaved. And we are going to pick up with C3 and what C3 does and other ways to get to C3 on Wednesday. So I will see you guys then.